Hello students, I'm Dr. Arpita here. A very warm welcome to my phycology class. Hope you all are doing great, and also, most important, hope you people are doing your regular, day-to-day revisions as well. Okay, so let's start our lesson today. The topic for today's discussion is, well, before we get into the topic, please like, share, and subscribe to my channel to get the regular updates of whatever I upload here. So the topic for our discussion is Oedogonium today. This is a bit long chapter, so I have divided this lecture into two parts. So this is part one of Oedogonium. Okay, yeah. So before we start our discussion, we must know the systematic position of Oedogonium. It belongs to the class Chlorophyceae. It's green in color, see? So you can understand it contains chlorophyll and therefore it belongs to Chlorophyceae, order Oedogoniales, family Oedogoniaceae, and genus Oedogonium, right? Please remember this systematic position. Without this, it's very difficult to study any genus. So class these four will suffice: class, order, family, and genus. Right. So Oedogonium is a freshwater filamentous alga that occurs in ponds, lakes, and stagnant water. So you will find this genus to occur in ponds, lakes, and stagnant water. What are stagnant water? See, you should pay attention to each little term that you get to learn, right? So what's stagnant water? Stagnant water is a water body or water that is not free flowing, right? That's stagnant water. The filaments are attached to rocks. Okay, so they remain attached to the rocks by what? By the holdfast and haptera. Those are fine hair-like structures that develop from the bottom of the holdfast. Okay, we'll discuss about those in details. It is also found attached on other algae such as cladophora. This is an important point. So this Oedogonium is also found attached on other algal species like cladophora, right? Some are epiphytic on aquatic plants also. Oedogonium terrestri is a terrestrial form and grow on moist soils, right? The young filaments are attached, but the older ones are free floating. Clear? Now see, the thallus is made of green multicellular. It is actually the thallus is made up of green cells. They are multicellular, unbranched, and filamentous in nature. Right? See, Oedogonium is unbranched, but for your knowledge sake, I'm telling you, Oedocladium is branched. Should know these, okay? Oedocladium is branched, right? Next, what do we have? See, All the cells of the filament are cylindrical except the basal cell and the apical cell. The basal cell is colorless, okay, and forms holdfast. The proximal end of the holdfast extends to produce finger-like projections which help the filament to attach on the substratum. It's somewhat like this. There are finger-like projections that help the filament to remain attached to the substratum. Okay? The apical cell is rounded or elongated in shape. If you pay a little more attention, these algal genus which you are studying, they are actually very easy. You just need to pay attention to these important key points. See, as we said, it is green, it's filamentous, it's unbranched. Look at the first line, green, multicellular, unbranched, filamentous. All these are matching. You don't need to trust me because I'm your teacher. I'm telling you, you will trust whatever I say. No, science is based on evidences. We should match the text with the diagram, isn't it? With the picture. This is a microscopic view of Oedogonium. You can see here, right? It is multicellular. See, so many cells are there. It is multicellular, so that is matching. Then it's green, it's unbranched, and it's filamentous. Okay. Now see. We'll start with the basal cell. It is the lowermost cell of the filament. Okay. The cell is long, gradually narrowed, and towards the basal end, it expands to form simple disc-like multi-lobed or finger-shaped structure. It looks somewhat like this. If I just magnify this and draw, it looks somewhat like this. Okay. The cell is generally colorless, which performs the function of fixation to the substratum and is called holdfast. Right? Next, we have the apical cell. It is the topmost cell of the filament. From the very word apical, you can understand it is at the apex, right? The cell is usually rounded towards the apical side and green in color. See, holdfast is colorless. Note these points. Holdfast is the lowermost one. What are the key points here? So for basal cell, it is the lowermost one, right? Then what? It has got lobed or finger-like projections or structures at the bottom, and it is colorless. Right? Here, what do you see? It is the topmost cell. The cell is usually rounded towards the apical side and it is green in color. Okay. See, this is how it actually looks. Look at this part. Look at this part. You will understand. Okay, now coming to the middle cells or intercalary cells. All of the cells in between the basal and the apical cells are alike. They look same. They are also called intercalary cells, right? The cells are longer than their breadth, that is rectangular in shape. How does a rectangle looks like what? It's like this, isn't it? The length is greater than breadth. Here also you see. This is the length. And this is the breadth. Correct? So the intercalary cells are rectangular in shape. See, if you just take a look at this, you will understand. See how long it is now? And see. Got it? This is one cell enlarged here. Towards the upper end of some cells, a ring-like structure is present known as the cap or apical cap. The cell with cap is called cap cell. The number of caps on a cell indicates the number of divisions in that cell. Right? Okay. Now come to the cell structure. This is very important. Please read each word with full attention. The intercalary cells are longer than their breadth and are cylindrical in outline. So these are the two key words that you have learned. They are longer than their breadth and they're cylindrical in outline. This is the structure. This is a very simplified diagram. So please practice this for your exams. The cells are surrounded by thick and rigid cell wall. Okay, this is the thick and rigid cell wall. The cell wall is differentiated into how many layers? 3 layers. An outer chitinous layer, that means the outer layer is made up of chitin. The middle layer is made up of pectin and the inner layer is made up of cellulose, right? So this part, if you draw, it is actually like this. It has got 3 layers to it. The outer chitinous layer, the middle pectic layer, and the inner cellulosic layer. Okay, just interior to the wall, cell membrane is present, which encloses the protoplast. The protoplast consists of cytoplasm, chloroplast, and nucleus. Obviously, this is not a primitive one, so it will definitely have one well-defined nucleus. See, it has got nucleus, it has got chloroplast, it has got cell wall, and chloroplast has got pyrenoids, right? The cells contain many small or large vacuole at the center and remain filled with cell sap. Along with cell sap, they also contain excretory products. The chloroplast is single, large, and reticulate. What is this reticulate? Net-like. Net-like. Okay. Single, large. I don't think I need to explain these terms. This remains embedded in the which remains embedded in the cytoplasm. Cells are uninucleate, obviously, see, one nucleus. And the nucleus is generally present in the center of the cell within the cytoplasm, or it may be eccentric. Okay. Or it may be eccentric. Now see, if you prepare these diagrams for your exams, just by looking at the diagram itself, you can write out, you can describe, while describing the diagram only, you will get to see that you are actually writing out a very good answer. Okay. So preparing the diagram is extremely essential. By the way, I had used a word eccentric here. What is this? What is the meaning of this word? It also means one-sided. So it can be centrally located or it can be eccentric. Okay. Now coming to the cell division in Oedogonium. This is very, very important. It's a little different from other algal specimen, right? Growth of the filament takes place through cell division. What type of cell division you are expecting here? Mitotic division. All cells except the apical and basal cells are capable of dividing through some of the cells of the filament, right? Okay. Now, pay attention to each word I say. I will discuss the various steps of cell division with you. Steps of cell division. Okay. Okay. So what's the first step? See, the nucleus, initially what happens, the nucleus becomes, or the nucleus shifts from the peripheral position towards the center, and then it gradually moves towards the upper side of the cell or upper half of the cell. So what happens here? You also write it down as I'm writing. Nucleus shifts from the periphery towards the center. Okay. And you can also write, and then gradually moves towards the upper half of the cell. Done? Next, what happens? A ring-like thickening develops towards the upper part of the cell. See, look at this. What did I say? From the periphery, this was the first step. From the periphery, it is moving towards the center. Then what did I say? A ring-like thickening develops toward a ring-like thickening developed towards the upper part of the cell. See, what did I say? This moves towards the upper half of the cell. If you divide this cell into two halves, see, the nucleus is not at the center. It is actually little towards the upper side. And see here, ring-like thickenings start to develop from these two sides. See, this is the ring-like thickening. So if we write this, how should we write? Ring-like thickening, ring-like thickenings from two sides develop towards the upper part of the cell wall. Right? Then what happens? The nucleus undergoes mitotic division and form two nuclei. That means what? Karyokinesis occur. Okay. Nucleus undergoes mitosis. That means what? Two daughter nuclei are formed. Okay. Now, at the end of the cell division, a row of microtubule develops and accumulates as a layer between the daughter nuclei. Okay. See this? What did I say? The nucleus divides mitotically to form two daughter nuclei. This is one, this is one. And in between, what happens? What did I say? After karyokinesis has taken place, a row of microtubule develops. Okay, let me write this. After karyokinesis, a row of microtubule develops and accumulates as a layer between the daughter nuclei. Develops and please don't use this type of and in your exam. Should write like this. Accumulates between the two daughter nuclei, forming a layer. Okay. See this particular layer, this layer remains in a floating condition, which actually will develop the future septum. Okay, this will develop the future septum, right? Okay. Now next, what do we have? Now what's happening to this ring-like thickening? What's happening to this ring-like thickening now? See. The ring-like thickening now gradually elongates. It first formed like this, no? This was the cell, right? It started forming like this, isn't it? Hmm? Now it gradually elongates. This thickening gradually elongates. So ring-like thickening gradually elongates and it splits the mother cell wall towards the apical region. Okay, elongates and splits the towards the apex or apical region, anything. See, this ring actually expands and forms a concave cylindrical structure. just writing, forms a concave cylindrical structure, right? See how is it forming? Got it? The ring material ultimately forms the cuticle of the upper daughter cell. So the ring material forms the cuticle of the upper daughter cell. Upper daughter cell. Okay. Next, the upper part of the ruptured mother wall. What did I say here? The mother splits the mother wall. That means what? The mother wall ruptures. Now what happens to that here? The upper part of the ruptured, see the upper part of the ruptured mother wall remains attached to the anterior end, to the anterior end of the new daughter cell, new daughter cell as a cap. So this is how the cap is formed. Okay, as a cap, right? This is how the cap is formed. Now see what will happen to that floating septum which I had talked about, that was formed by the microtubule layer. The floating septum will gradually go up. Where will it go? It will go up to the base of the future cell. The floating septum goes up to the future cell. Okay, that is what? That's what? That is at the top of the mother cell. Mother cell at the ruptured end. At the ruptured end, and it becomes fixed. It's no longer floating, right? And it becomes fixed. Later, it develops into a mature cross wall. Okay. Later, it develops into a mature cross wall. Clear? Understood? Try to imagine what you read. Once you develop this power of imagination, you will never forget what you are studying. Okay. Now see, new side wall develops between the cuticle and the plasma lemma of the upper cell. Thus, the two cells are formed. Okay. So at the end, what happens now? New side walls develops between the cuticle and the plasma lemma. Cuticle and the plasma lemma of the upper cell. Okay. Thus forming two cells. So this is how we come to an end of our cell division process. Okay. So I'm ending the first part of the lecture here. So let's just quickly recapitulate what we have learned today. We have learned about the systematic position. Then we have learned about its habit, the thallus, the basal cell, the apical cell, and the intercalary cells, the basic cellular structure, and the mode of cell division. The steps of cell division we have discussed in detail. Okay, I hope you have enjoyed the lecture. If you find it really useful, please share it with your friends and subscribe to my channel. Stay connected for the second part of the video. Till then, goodbye.